I'm the Vice Chancellor here at Te Whare Ōrākei Link University, and it's a real pleasure to have you all here today to welcome the winners of the 2024 prestigious Prime Minister's Science Prize, and in particular Linda Johnston and David Hugh to present here today. They're going to talk about Endophyte, and I had a little journey about Endophyte that I was just talking to Dave about previously. When I did an honours project here at Lincoln in 1990 with Richard Lucas over there, I did a project which was around the impact of Endophyte on grazing and crash in sheep, and that was the old wild type Endophyte. And at the time, it was just at the emerging stage of novel technology around Endophyte was emerging, and there was the thing around Endosafe. I think we might park that at some place, and as to what actually happened with Endosafe and that particular Endophyte. And I went to the UK at that point in time, and going from a country where Endophytes actually mattered to a country where no one really cared in any way about Endophyte and talking about it was an interesting position to come to. But today we're here to celebrate the outstanding achievements of the AgResearch Endophyte team, who were honoured with the prize for the decades of work, their pioneering work around advancements of Endophyte technology. Those new Endophytes that were beneficial, giving the characteristics to enhance perennial ryegrass plants in particular, and remove some of the poor animal health effect. This is real breakthrough stuff that has contributed hugely to New Zealand's agricultural sector and our economy. And we're here to acknowledge today the remarkable journey, one of dedication, innovation, and real world impact. Please join me in welcoming Dr. Linda Johnston and Dr. David Hugh. Thank you.
Good afternoon, everyone. It's a real honour to be here today and talk to you about our journey around winning the Prime Minister's Science Prize. And as you can see, it's a team of us up here, and not only is it us scientists, but there are many important researchers and staff who've been involved in this journey with us. And as I said in my acceptance speech, we are standing on the shoulders of many giants, and those people have come before us over decades of research. And I can see we've got one of them in the audience today, Lester Fletcher. Right. So, Epicloe endophytes, from science to commercial impact, and that is really our story. So David and I are going to do a bit of a joint presentation today, and a number of topics that we're going to cover. A lot of it is quite historical, and as we go through the talk, we'll go through our journey towards the more recent science that we've been doing, which includes gene editing of endophytes, as well as how we've managed to get endophytes into cereals. So we've been very fortunate to be part of a multidisciplinary research team, and I believe this is one of our strengths, and it's all around plant-microbe interactions and to understand endophytes specifically and the role they play in developing resilient and productive pasture systems. The science that we've been very fortunate to do from a combination of government and industry funding has really allowed us to have a full pipeline from that very fundamental science, trying to understand how do endophytes, what do they do inside their plant host? Do they just sit there and do nothing, or are there molecular signals, which we in fact know that there are? All the way along to how do they transmit through the life cycle of the plant? How do they get into the seed? And a lot of the applied science has been absolutely critical for the commercialisation of these endophytes. We have diversified, and we don't just work on these endophytes of grasses, which are known as epicloe, but we also now are looking at endophytes for the carrot seed crop, for other diverse forages and things, and we even work on bacteria that are also microorganisms that live inside plants as well. So this is the endophyte science team and all the different disciplines that we use. So you can see it's quite diverse. And we also have our partners, Fiji Rights and Feeds and Grassroots Technology, and John carrot has played a major role in the commercialisation of these epicloe endophytes of grasses. This way. Press a button. Oh no, here we go. So first of all, I want to start with a bit of a definition in case you're wondering about what an endophyte is. And it actually comes from ancient Greek. So endo means inside and phyte means plant. So it is a microorganism that lives inside a plant, can be a fungus, bacteria, or even a virus. And it's been coined and recognised by De Barry in 1866. So these, and in fact, the beneficial effects of these microorganisms have been described for more than 200 years. So it's really quite an old thing, but it's really the microorganisms that cause disease that usually get all the attention. So the difference between an endophyte and something that causes disease is really the difference that an endophyte is within a plant and it doesn't cause disease. And in fact, we find endophytes everywhere in all plant species, and they all have different roles. And the beneficial ones, they're really improving plant fitness. So beneficial endophytes for sustainable do provide sustainable solutions for agriculture. And really today we'll be focusing on the role of these endophytes around controlling pests in our pasture systems. But they also control diseases, and endophytes in general can do all of these different things on this list. So they're really amazing microorganisms. So now a little bit of a focus on the star of the show. So epicloe endophytes, or also colloquially called grass endophytes, are a really prime example of a beneficial endophyte. And we've written, the team and I have written a number of reviews about these organisms, and I'd just like to point you to these transmission and SEN micrographs. And if you take a cross-section of a tiller, in between plant cells, which is where these hyphae live, they are attached to plant cells. And here you can see a hyphae, and in this image you can see in between the plant cells, there's a plant cell here and a plant cell here, so it's your hyphae. And I'm going to talk a little bit more about how they grow in a moment, hopefully. So what you can do in the lab is you can label them with green fluorescent protein, which makes each hyphae glow green. So these endophytes, they're obligate in nature, which means they're not free living, they're only found inside their grass. But in a lab, you can actually culture them on a petri dish. Now, in a plant, in a grass plant, you only find them in the upper part, and they're often really just around this lower basal area of the plant. And if you take a cross section, which we've done here, and this is blown up, you can see all the different leaf shoots and the emerging leaf blade of the grass. And isn't it incredible, all the green dots that you see? So grass have all these hyphae, so they're coming towards you, if you like. So there's a lot of endophyte within a grass, and you can only see it using a microscope. So what is it that they do? A lot for our agricultural system, which we'll get to. First of all, I just wanted to point out that in this schematic diagram that the grass and the endophyte growth matches each other, or you can say it's synchronized. So the reason that we're interested in understanding about this is that when it comes to a product, the endophyte is actually sold within the seed. So it's a seed technology, and it starts with the endophyte actually being right inside the embryo of the seed, which is the living component, and you need a viable embryo to get a plant. And what happens is that endophyte will then grow into the new tiller, and then it grows systemically up the stem. And actually, because those hyphae are attached in between the leaves, as the leaf extends, so does the hyphae. And then when the leaf stops growing, that fungus also stops growing, but it still makes its compounds, which is really quite unique. So eventually that endophyte is carried into the seed bed, and it actually needs to infect every single seed. And it's also needed for commercialization to have highly infected seed. So we do a lot of work trying to understand how that endophyte is happy in that plant, so understanding the compatibility. And what we also know is that the endophyte is actually not in the pollen, and there aren't any spores, so it's a completely contained system. We also do a lot of work understanding the viability of the endophyte in the seed, which is another important component for developing technology. Now, David is going to talk a little bit more about this aspect. Did you want this?
Thanks, Linda. Now, from my talk, my slides, I want to bring in a bit of the local aspects and feature some of the people more. So when we did the talk in Palmerston North, I talked more about the researchers that are based in our Palmerston North campus. And we're in Lincoln, so I want to feature some of the field work that's been done here and some of the people that have been done here as well. So it is about smart people thinking together and making this work. And we next out in the landscape, perhaps beyond the Lincoln area, have a look around and think about the plants in our landscape. Now, this analysis was done about 10 years ago. It's probably still about right, where these are the three most important, economically important species in our landscape. Unfortunately, they were all introduced to New Zealand, none of the natives. But think about the pine trees, pine forests that we may see distant. That figure is probably a bit bigger now with the recent plantings. But look at the value of ryegrass and clover here, particularly ryegrass. So it's about $15 billion worth to the economy each year. We just think about Linda telling us how these endophytes are in grasses, particularly ryegrass. We've been tackling a really important pasture species or economic species for New Zealand. I just want you to take that concept as we go through. Right, there's some pros and cons of endophyte. And this is why it's been a real journey of generation of some decades of work. So on the positive side, endophyte is really good for plant persistence and production. And I've used this here as an example. These are our Lincoln AgResearch farm, a couple of 100 metres that way, which Lester and the team would have been using. And see, this is the same cultivar, either with endophyte on the side or no endophyte. And you wonder why the differences there, or it comes down to these insects. Right. These around this pest protection. So we've got here the Argentine stem weevil. So in those days, Argentine stem weevil was a major insect pest in New Zealand pastures. This would be in the 80s. And you can see with endophyte, it's maturing the stem weevil. But here, they think it's just ice cream, it's a pest. And it's not that. And it's actually not the weevil so much of a problem. It's actually it's larvae that develop in the very base of the plant. You can see that they eat out the growing points here. And when they're finished with one growing point, they move across to the next branch or tiller and eat that. You can see they're only a few millimetres long. You'd hardly think they were devastating, but look what can happen. So that's a really, really important advantage of having endophyte. And particularly in New Zealand, we keep finding that insects are driving these factors, but we also know there's disease resistance and other factors going on here as well. Right, that's the positive side. But on the negative side, there's some of them are harmful to livestock. And again, I'd like to feature the work here. These are photographs from Lester here in the team. Initially, it was all around linking endophyte to ryegrass status. And for those of you who don't know, this is not a normal position for a sheep to be in, and I'll talk more about that soon. This is probably the slide or the photo I want you to focus on most of all. And you can imagine when I do talks in Australia to an audience, the jokes about New Zealanders and sheep come into play, and they have a really good laugh at me. But this is a fantastic illustration of the impact that the standard or toxic endophyte can have in New Zealand. So yes, obviously forest ryegrass diggers, but these two lands here have been grazing here in our Lincoln farm, and pure ryegrass, and look at the size difference here. The endophyte is a penalty on these lands in terms of growth rates. Look at the backside here, so we're getting these feces accumulating the wall, getting dags. We end up having blowflies coming along, laying their eggs in here, and they get developed and eat into the fish of the sheep. So you can imagine it's very distressful for the sheep and for farmers. And also there's a factor of exacerbating heat stress. Now, so I was talking to John McKenzie a few months ago, and John was telling me about how temporary sheep farmers just couldn't get the production of their lands during the summer. And it's typically called summer or autumn ill thrift. Now, ill thrift can be due to lots of different things, but it's definitely due to this toxic standard endophyte. So yes, ryegrass steg is important, but look at these production issues here. If we're in the white cover, they showed the dairy enzyme showed a 9% penalty in terms of milk production. So sorry, I'll just go back there because there must have been a huge dilemma in the 80s. I was just finishing Lincoln University at the time. I was about 8 years behind you, Graham, I think, in terms of graduating here. But what would you do from a farming point of view? Yeah, look, you needed endophyte for ryegrass persistence, but you were going to have these problems. Take endophyte out, we're going to have lovely, healthy growing lands, but you may not have anything for them to eat. So it's a real dilemma, and really the industry did go with what was called high endophyte. So you had persistent pastures, and you had to put up with this. And I'll talk about solutions to that shortly. Right, this is going to be fun games for me. Right, sorry for that. So, but I'm going to say a little bit more about that. So it's classified as a neuromuscular disorder. Essentially, for humans, if you've drunk too much alcohol and you start to stagger home, that's what these sheep are doing. And the key thing here, it's amazing, it occurs in a wide range of animal species. You name it, they seem to suffer from it. Some of these we shown experimentally. So here we have camels. Experiment was done in the Middle East, and it's because they were getting straw being imported, ryegrass straw, and it's causing ryegrass seeds and camels. Experimental work with alpacas in Australia. Of course, the sheep work was done here in New Zealand at Lincoln. This one up here with cattle was experiments we did in Manawatu. And probably the most interesting one is the white rhino that you can see down the bottom here. Now, this is the white rhino in the Auckland Zoo. We were contacted by Dairy NZ, who had been contacted by the vets in the Auckland Zoo. And they thought, animals are behaving unusually, and they thought it could be ryegrass stems. So they gave us some of the hay, and well, you analysed it. We thought, yes, there are some ryegrass toxins there, but not very much. They removed that endophyte hay from their diet, and the animals completely recovered. So my guess is this might be the most sensitive species on the planet, but we're not going to run an experiment on such rare animals. And it's also been reported in the zoo in Australia and others in Australia. Go back. Right, look at Australia. This is what can happen in Australia. And here the syndrome seems to be just sort of all mixed up, and they term it real ryegrass endophyte toxicosis or toxicity. And what can happen in some years, you can see massive livestock losses, which we don't see in New Zealand. And sometimes simply sheep can be found dead in the paddocks, and other times the sheep will drown themselves in these dams, which are used as water sources throughout the paddocks. Even when farmers fence off these areas, the sheep will still seek water. So this is major. This is like $100 million problem a year in Australia. We don't see these widespread toxicity or death problems in New Zealand, and we still do not fully understand what's driving that. We're going forward. I'm going to have to get you to do this one. You seem to do the right things. Right. So let's get back to New Zealand. Here's a paper, 1981, New Zealand Veterinary Journal, called a correspondence. It's only a few pages long. That's by this the picture here from DSA in those days, and Ian Harvey, who is still active in the field. And they published this paper around linking ryegrass staggers and endophyte. And this is a section from their paper here that they believe the ryegrass staggers is linked to this fungus, a possible cause. And this, I'm not too sure whether you and Ian at that time really recognised the magnitude of your findings, but it was definitely linked to it and really just solved a huge mystery about what was causing the staggers. Researchers have been working on this for 50 years, and Lester and Ian solved it. So Lester, and along with Lee Sutherland here at Lincoln, was able to set up this system with small paddocks and testing here, and did a lot of work, not only to identify the ryegrass staggers issues, but they also identified and worked on heat stress. In this case, the sheep were brought into a tunnel house to elevate the temperatures. And the lady in the back there is actually Lester's wife, Carol. So it was a family affair there. So Carol did a lot of work on this as well. And of course, those animal productivity aspects that I showed you too. So yeah, it really, really can keep part of our research, the work that's been done here. Right. Okay. Another, I'm a scientist, I need to show you some data, so I'm desperate for doing this. This one here is actually, you'll see on the bottom here, Grant Edwards, which is the same Grant Edwards that's over here. And Grant stole my thunder at the start because he talked about his thesis work that he did in 1990, an honours thesis. And you did it with Dick Lucas, and I did my honours project with Dick Lucas in 1982. So what Grant did here was he had sheep grazing these pastures for a certain number of days on here, and you see he measured the height of the ryegrass. And I'll show you soon that it had ryegrass either with no endophyte or with the standard toxic endophyte. And Grant measured it over time. Then you can talk to us. Hey, can you click that on? Or no. So what we can see here over time is that the sheep behave differently. So with the standard toxic endophyte, they decided, oh no, I'm going to stop grazing. I'm going to go to a certain level. But when there's no endophyte, they just keep on grazing down and down. Now, from a ryegrass endophyte point of view, the endophyte wants to help that plant survive. So basically, it's protecting it from overgrazing. When you don't have endophyte, it could be overgrazed and be penalised in terms of its regrowth. Now, that has implications for our grazing systems. And I think, Brian, you were the first one to document this for ryegrass. It became quite evident in tall fescue endophyte in America. And I think even today, we still don't recognise this enough and think about it in our pasture system. So, not only AgResearch has been involved in the story, but other people as well. Right, and Grant, this is for you. So we can see that this is not an experimental situation. The same sort of grazing preferences can come into play. And in this case, we've actually selected endophyte on the right-hand side. So next slide. Right, so you can see what are the potential solutions to addressing these animal livestock toxicity. And first of all, we can simply avoid having perennial ryegrass at the endophyte. There are other grass species that can be used. We can dilute it with clovers, herbs. We can use supplementary feeds. There are also other options of animal breeding, biocontrol. New Zealand's been very successful in that, and people like Steve Goldson here can talk about that. We've got a very good track record in New Zealand. Changing grazing management as well. But really, out of all these options, selected endophytes have been the winner. And you can see AR37 endophyte, for a $12 million research investment, it's yielded us $3.6 billion benefit to New Zealand. Just by having the right fungus inside that plant, a fungus that Linda's showing us that you can't even see from the outside. So it's not the only solution, but it's been an incredibly successful solution, and that's why Linda and I are here today representing the team in the story. Right. So I'd like to go back some more to the people, and we'll go back to Palmerston North in this case, and I consider these two gentlemen here to be the forefathers of selected endophytes. So Gary Leach is a fungal expert and Brian is a chemist. The concept is, and it proved to be true, that there are different strains of endophyte. So the strain that was all through our ryegrass in New Zealand was essentially the same. And it was all toxic, it was all bad news for our livestock. But overseas, there was a lot of diversity, and that's what we've all used. And there are good strains. And by good, what I mean is the good for a partial setting is that it's non-toxic to our farmed animals, but still provides that protection against the pasture pests. Now, consider one of the key things that Gary and his team did was that they were able to move the endophyte from plants which were collected in the Mediterranean basin, great place, but those plants weren't adapted to the New Zealand environment or Australian environment or somewhere else in the world. So Gary was able to culture in the lab. You remember before that Linda showed a culture, so that's in the laboratory. They were able to take that and inoculate it into seedlings of New Zealand cultivars or tall fescue, American cultivar, Australian cultivar. And somebody like a rock, she's a rock star in our lab for the number of inoculations that she can do in a day. So myself, I was brought up as an agronomist, a plant person, but this was key to getting, once these good insights were found, we can rapidly get them into modern cultivars. What we had is a pipeline of discovery and development commercialization. So we bring these seeds in from overseas, we screen them for endophyte, chemistry and genetics. The ones which actually which we think are good, they get inoculated, like I showed you before, insect tested, seed production, and particularly a lot of work done here by Phil Rolston and his team. Pasture testing into the animals, again, linking the animal testing has been critical, and then right through into product for New Zealand farmers. All right, just to explain about the different endophyte strains, because that's been critical here. So Alison, who has been our lead entomologist for the last 30 years and years, just retired this year. She uses this slide here to explain to farmers the context. So you can imagine we've got two ryegrass plants here. The one on the left is endophyte free, so no endophyte in it. The one on the right has standard endophyte, the one that's widely spread throughout New Zealand in the old pastures. But we look at these, we load on here, these are the 6 major insect pests of endophyte-free ryegrass in New Zealand. And it's quite an interesting story, and again, it's another one that Steve Goldson can tell us in another lecture. But this is from African black beetle, so it's from Africa, Argentine stem beetle in South America. And then we have a rootated here, which comes from the Mediterranean. And the other 3 here are New Zealand native insects. And they want to be bought our European pasture species here in Southern fertilizing. These native insects just love it. There's ice cream again. They just flourish. It's actually not a very good place, a hard place to grow ryegrass in New Zealand if you don't have endophyte. Let's just add in when you've got standard endophyte. You can see some of these insects pictured, they disappear. And some of them, you'll see what I have here, some of the pictures get smaller as well. So the standard endophyte has some control of some of these insects, but not complete control. And that's why the original photograph I showed you of plus and minus endophyte, the huge difference with the stem level, is basically due to these insects. Now, the first major endophyte that we released was AR1. And you can see it's not quite as good at controlling insect pests as the standard endophyte, but it's certainly better than having endophyte free. And we thought it was all over then, and we'd sort of done our job. But another endophyte was discovered, and it was AR37. And you can see, if you can, from down at the bottom there, we only have one major insect pest that it doesn't control. I never thought we would ever get to this point. And now, just more recently, you see other seed companies are starting to find their own AR37 equivalents, and that's coming out on the market just now. So it has proved to be a very effective endophyte. And remember that $3.6 billion value term. So that's the good side of having endophyte, is pest protection. But let's lay on top of that the animal aspects. So start with endophyte free. But no endophyte, it's safe for animals. Compared to the standard endophyte, we get ryegrass figures, animal production losses, deaths, all sorts of problems here. And that's the one that's widespread in the wrong pastures. AR1 is considered animal safe. AR37, not quite animal safe. It still has some ryegrass figures, but we don't have any of the heat stress problems or loss in animal production. Not quite the perfect endophyte, and Linda will talk more about that later on, about our next phase or stage of endophyte coming through. Right, I'd like to take you back to some of the unique findings that have come through from this work. This is in the Marama 2, and we have these plots here, which in the autumn, we found that some of them looked quite a bit greener than the rest. Why? We couldn't see stem weevil, couldn't see the usual insect pests there. But what we did find was this root ache. We never knew this was a problem in our pasture. We never knew that endophyte was actually doing it in control until AR37. So AR1 and other endophytes still controlled us. But when we had AR37, it was controlling the root ache. We were getting better production and persistence. So that was a real surprise. So back to a Lincoln example, here at a paddock scale during the winter, what was driving these differences? It was, in this case, it was pasture mealybugs. We didn't know endophytes controlled pasture mealybugs. This insect had been previously described in New Zealand, but wasn't known to be related to the sort of devastation that we could see some years, and that endophyte controlled it. And it was through some pretty smart observations from Chris Pennell, and some of you that know Chris Pennell, he's a very observant character, and working alongside Phil Rolston. Phil's actually in a lecture on the campus at the moment, will come in shortly. These guys have been really key for the agronomy work here and the seed production in this environment. Right, I could almost tell you the same story for other grasses, so such as tall fescue and meadow fescue. In this case, tall fescue in New Zealand didn't have a toxic endophyte in the cultivars which we had for our own. Did in America, but not in New Zealand. So our challenge here was we found completely animal-safe endophytes, and you can see the productivity advantages that they give. Now this is actually up in Northland, where we've got black beetle as a major issue here. So we're in the upper North Island. It's a major advantage to have an animal-safe endophyte in your tall fescue cultivars. Right, just going back to ryegrass, I've shown you the effects of different strains of endophyte, these selected endophytes. We haven't really found the perfect one. It's always a bit of a balance. If you get more insect control, we tend to have some downsides in animal effects. If we have very safe endophytes here, we get downsides on the insect control. So there's benefits and risks to this. Again, just emphasising how AR37 was a game changer, where its insect control really took the balance in favour of this endophyte, but we still had some mild animal health problems here. And really, the success of this endophyte, 65% of ryegrass feed sold now has the AR37 endophyte in it. So farmers have voted with their choice, and it's been really successful. Still not the perfect endophyte, and again, that's what Linda will talk about soon. And Linda, I think that might be. I'm going to hand this little beast back to you.
Thank you.
All right, so just before Linda starts to, I've shown you the how endophytes affecting insects and animals, but I haven't told you the reasons why. And that's where Linda's going to talk about the chemistry and all that bioactivity. So we understand a lot of the reasons why. Well, Linda.
Thanks, David. So on a very high level, I am. So we do know that endophytes make secondary metabolites. So these compounds are known as alkaloids or secondary metabolites, and a number of them have been well defined from a number of researchers around the world and not just from us. So very briefly, we've got a number of toxins. So Lolitrin B is an indole diterpene compound, and that's the compound that causes ryegrass daggers. Also, there's an epoxyxanthin compound, which is similar to Lolitrin B, which also causes ryegrass daggers and is in AR37. Ergovaline was discovered after Lolitrin B, and that causes heat stress in grazing livestock. And we've also got a number of really good compounds that we know about, such as pyramine, which deters Argentine sting weevil, so it's got a big happy face there, and our lowlands, which actually are both insecticidal and a deterrent, and they provide a really wide spectrum of protection against insects. So you just see a compound, but actually they are synthesized by a very complex pathway where you have a number of genes that slowly add these decorations and make these really complex compounds. So they're not simple pathways at all. So an understanding of those pathways has been really pivotal in on our search for selected endophytes that are better. And we've actually used that knowledge to screen endophytes that we bring in from around the world. So that's part of our endophyte pipeline. So just briefly starting with AR1, now it doesn't make ergovaline, but it provides protection to Argentine stem weevil, and this is because it makes pyramine. So if we're interested in, well, there are certain insects in our pastures, which David has described already, and we obviously don't want to have Argentine stem weevil. So when we're screening an endophyte that makes pyramine, then we know that we're going to have protection. So we can use chemistry as a very quick tool to understand what we might see with our endophytes. But AR1 was a technology breakthrough. It was commercialised in 2001, prevents ryegrass staggers, doesn't cause heat stress, reduces fly strike, and we saw an increase in live weight gain of more than 10% and an increase in milk solids of 9%, as David mentioned before. This is just an illustration of how we actually deliver an endophyte to farmers, in this case AR1. But we inoculate a proprietary cultivar with the endophyte. We check the alkaloid profile and make sure that it doesn't have anything it shouldn't have in it. And we also need to make sure that the endophyte is found in that seed. Otherwise, farmers don't get a product that's useful to animal safety is also the other aspect, and the agronomic performance. So those are kind of all the key steps. And similarly, that was done for AR37 and the new endophytes that will be commercialised. So AR37, I quickly mentioned these epoxyxanthin. So they have these compounds which provide tolerance to insect pests, but they can also sometimes cause ryegrass daggers, and that's why AR37 isn't a perfect endophyte. And I guess the most significant economic value has been AR37, which has been estimated to contribute that $3.6 billion to the New Zealand economy through the life of its patent. And there you have a bag of seed, which farmers will select. 65% of our proprietary cultivars have that endophyte in it. Now, this is just a timeline of the endophytes that have been developed and some of the history behind it very quickly, which you've seen today really from David's talk. So I won't go through this too much. But in the 80s, we had the linking of the insect with ryegrass daggers and the presence of those lolitrin compounds. And, you know, we tried endophyte free, but it wasn't persistent. Then there were these toxic lines. We didn't actually know they had ergovaline in, but having serious heat stress in animals was obviously not ideal. And then we had AR1, which was really the first breakthrough because it was completely animal safe, and it provided resistance to the first major pest that we were aware of that was a problem in our pastures, which was Argentine stem weevil. Then the Endo5, which still had some ergovaline, but it was a lot, is nowhere near as toxic as the original line. And the problem you have is sometimes these mammalian toxins are also insect toxins too. And so I'm going to talk about how we are improving this through gene editing in a moment. AR37, the next big step change in 2007. And that produced a minimum of a 12% increase in dry matter yield. So from a plant plant productivity and in animal productivity, it's really fantastic. In 2008, we started the cereal endophyte program where we thought, well, wouldn't it be amazing? These endophytes provide such wonderful control in grasses. Cereals are grass. Could we put them into cereals? Have to tell you, it's really not that easy. But David will talk about that briefly. We've also tried to take endophytes from tall fescue and put them into ryegrass, but there are compatibility issues. And so I actually started as a postdoctoral fellow working on the cereal endophyte program, trying to understand the compatibility between endophytes. Anyway, then we had some success in rye, which is the Kali cereal, because it's an outcrossing species, and we're more successful in getting endophytes in there. And we've had some success with wheat. And more recently, we filed a patent for gene editing, and I'm going to talk to you about that program next, I believe. Right, so gene editing. So this is some of the future, and it's really around improving both our insect protection, thinking about the fact that we've got climate change, we've got insects that aren't sticking to their normal territories, they're moving further south, etc. So we might need to have wider pest protection and protection against different pests. There might be new pests that might be a problem in the future. Plus, animal safety is obviously really important for our farmers. Current commercial strains, so we've been searching for new genetic diversity for a very long time, and what we find is that most of the endophytes that we bring in, we've already seen them, and nothing can compare to AR37, or they're toxic. So most of them are toxic. So it's really difficult. We've already picked the best off that tree. So the variation in the strain with respect to that chemistry, the right chemistry, is very narrow. So how could we create new genetic variation? Well, we can through gene editing. So I'm going to describe that to you now. So we started actually 6 years ago with an NB partnership bid. And so this was NB along with Grassroots Technology and PGG Rights and Feeds. And we're wanting to provide those benefits that I mentioned, as well as even new options, because endophytes can deliver a number of enzymes and proteins that could have new functions that maybe we haven't even thought of. First of all, I just need to tell you a little bit about the technology that we're using. So gene editing is kind of known as a new breeding technology, and really what it is, it's a biotech-based method which can rapidly and precisely improve plant traits. And I've got a little diagram over here, which so a lot of the methods involve things like CRISPR, which you might have heard of, and basically we've seen an improvement in development of these. We've got more precision, they're more feasible to use, and it's a bit like our phones. So, you know, starting off from when you used to have a phone, you just made a phone call, to the first cell phone, to now these phones that do everything that you can imagine. Well, that's a little bit like CRISPR-Cas9, and I have to say, CRISPR-Cas9 has actually improved our knowledge of endophytes and the compounds they make, because we really couldn't use molecular biology with AR37 before CRISPR-Cas9 came along. We couldn't delete any genes, we couldn't add any genes. It's really difficult to understand how it functioned and what it did. So these breeding technologies can enable you to turn a gene off, which is a mutation or a knockout. It can help you to change the sequence. It can enable you to fix a sequence. There might be a mistake. And you can insert new genes, which are genetically modified organisms or knock-ins. All of these new breeding technologies, including CRISPR-Cas9, enables you to precisely target a specific place in the DNA in your genome. And they all do it. They're all nucleases or enzymes that create double-stranded cuts in the DNA. They're not all the same. So all of these enzymes, they can either be used to basically make a double-stranded cut, and the cell will try and repair it itself, and it makes a mistake, and that's known as an SDN1 or a site-directed nuclease 1, or you might use a DNA template, which might be copied, and you can add genes or replace segments of DNA. And so the reason it matters is with our new gene tech regulations coming online, if they match Australia like they're supposed to, when you do these edits, they will no longer be regulated as genetically modified organisms. Whereas these ones, because you're inserting new DNA, they will be considered genetically modified organisms. So we have developed SDN1 endophytes, and oops, just sorry, must have hit the wrong button. So we have developed a number of SDN1 endophytes. We've actually managed to take some of the commercial endophytes, particularly I talked about the commercial endophytes that made lots of ugabaline. Well, we can actually get rid of that ugabaline now, and we have. We've also looked at one of the other endophytes that makes ugabaline and been able to get rid of that. And we've been able to take AR37 and we've been able to manipulate that pathway so that we could switch off all the toxins, or we could just keep some of those that might provide some insect protection. And we've evaluated them. And right now we actually have a number of SDN1 endophytes in Australia, New South Wales and Victoria, and we're running agronomy trials. But if the gene tech regulations come out like we expect them to, like Australia, we'll be able to test them here in New Zealand, which is really important because there's endemic pests to New Zealand, and we really want to see how these endophytes perform. Will they be better? Will they be that more perfect endophyte? That's the question. The other thing that we can do is we can add new genes, and we can add them from other endophytes. So sometimes you've got a kind of a good endophyte here, but it doesn't have these features. So maybe we could take the genes that encode the compounds of interest and put them in that one. Or, for example, we could even deliver anti-methanogenic genes, so we could influence the amount of greenhouse gases that our animals produce. So we could have an anti-methanogenic compound being expressed in the endophyte, which is in the grass. The animals eat the grass, and that will actually reduce the methane from those animals, for example, which is kind of cool, very futuristic, but it might happen. I just want to illustrate this in a little bit more detail very briefly to show you some of the complicated science, just as an illustration. So these epoxy anthrogens I've mentioned, you know, they can cause ryegrass staggers at times. They're short tremors, unlike Lolotrim B, which is a long tremor. And they also provide resistance to purina, which is really a big issue down here in Canterbury. And when I told you that these compounds are made by complex pathways, well, this is kind of what it looks like. But using gene editing, we've been able to elucidate what this pathway looks like, and all the different genes along the way, and all the different compounds that are made. So we know that these ones here at the end are the real toxic compounds. So we can actually completely get rid of them by doing some gene editing, and we can accumulate some of these intermediate ones. And we've discovered that some of these intermediate compounds are still providing really good insect protection, which is really valuable. And we can get rid of this whole pathway so we can actually understand about the other compounds that might be in AR37. So it's a really valuable tool. And that's just some of the chemistry, just for fun. Right, the last segment is cereals.
Yeah, I'm going to take.
And you're going to have to take it fast.
Yeah. So this is a much shorter story. Could be another hour lecture for fun. And please bear with me as I fashion the pointer. But this is about another 20-year program, and the team thought, well, can we actually have these endophytes to be of benefit to cereals? And they're not in our modern cereals, but they are in the distant wild relatives. And what we have found is it could be transferred into cereals, and the answer is yes. Were there animal safe ones? The answer was yes. And could they increase the pest and insect protection of the cereal plants? And the answer is yes. So you see, it's a very short story, very similar one for grasses. And I just want to feature a couple of people. So Richard and Wayne, who are both actually based in Palmerston, have been really passionate about this. Wayne actually completed his PhD within the early days of this program. So he's a real expert in this space, fascinating to talk to. One of the key things that the program found here was the critical importance of plant genetics. So these two rye plants here have the same endophyte strain, but look how different their stature is. And this is all due to different plant genetics. Obviously, we want plants with endophyte to look like this and not like this. We know it's really important in the pasture space, but even more important in the cereals. And to help solve that problem with wheat, we were able to team up with Hasashi Sugimoto in Japan. And he had key wheat genetics, which we were able to use in collaboration with him and his glasshouse system in Japan to find the right genetics. So part of our AgResearch success has been our success with our collaborators. Never do science all by yourself. And what we have now, this is at Lincoln, we have Lisa Wood, who's doing her PhD here, based at Grassroots at Lincoln, jointly supervised, I think, Rain, if you're still here. Rain is one of the supervisors, as well as the University of Adelaide. And Lisa's already recording how the endophyte and the rye plant here is reducing this rust disease on here. Where you've got endophyte infection, you've got a lot less rust. So it is going to deliver benefits to the cereal program, to the cereal world. And Linda, I think this is back to you.
All right, so this is our last slide, and I just wanted to summarise that hopefully you agree with me that these endophytes are amazing biological controls, and they provide a sustainable solution for controlling pests and diseases. And we do have them now also in cereals. It's still a bit of a journey, but we do have in Australia an Australian breeding company who are trying to get them into their elites or back crossing. So that's a good start. So, moreover, some of the newer technology like CRISPR is enabling us to really understand more about how these endophytes work. We didn't really talk today about how the endophyte talks to the plant host, but we also do a lot of work in that space as well, which is really critically important for future products. So these endophytes make compounds, and it's really understanding those compounds and manipulating them that helps us be able to change the gauge between animal safety and insect protection. So with that, both David and I would like to thank you very much for coming today, listening to us, and we'd be very happy to take any questions.
I'm going to start because between AR1 and AR37, that by my calculations, there's 35 in between. It didn't work. How did you deal with setbacks along the way?
Oh,
You start with it?
yeah, you start that one, I think.
Well, Grant, I'm just expecting you to ask them how.
Yeah.
Well, I think it's perseverance when it comes to research. You expect a high failure rate. It's a wee bit harder when you actually put your foot out there into the commercial world, and you've got commercial companies are relying on your technology because they're going to lose money. They've got shareholders, okay? They don't want too much risk in their life. Researchers are more useful. So look, it's been difficult, and it's required a perseverance and understanding from a science point of view that out in the community, the industry and the public can not always understand the risks of science and how to handle it. That's my rough answer, guys. So we're going to have Linda
Well, any kind of problem, that's what scientists do, right? They solve problems, and in some ways, you know, there's no need for science if there aren't problems in this world. So it's also an opportunity, and it's an opportunity to learn about the system and to understand what it is that we need to do differently. And so, for example, there's been issues in the past before I was even here, where an endophyte, and it's this whole thing between continental and Mediterranean endophytes. And so what we do know is that these compounds that endophytes make, sometimes they can be silenced, and you change it to a different plant host which has a different background and environment that the endophyte lives in, and that might switch on and off compounds. And so it's possible to have some unexpected events. And so really, I think we've got some of our partners here today, PG Rates and Feeds, which is really lovely to see you guys coming along. And I think it's been a really perfect partnership. We've also been developing a relationship and evolving, and understanding what it is that you guys need, and you guys also understand that we're quite passionate about understanding how things work. So it's kind of finding that balance between what is the science that we need to do that will help not only the industry partners, but help New Zealand farmers, because ultimately David and I have to do science that will benefit New Zealand. And so that's really what we're striving to do, is to make sure that farmers get those benefits.
Yeah. If I could just add to that, and I think you touched on it, David, but it's actually managing expectations. And I mean, you mentioned Grant, Endsafe. There was nothing wrong with Endsafe. Actually, I mean, it was what was wrong with it. It was marketed with the expectations beyond what it could have done. And so actually, industry was denied an opportunity 5 or 6 years before it could have had it. It was a dirty endophyte, so it wasn't perfect, but it could have been commercialised. But we were looking for the perfect solution, so that the royal we. So then the expectation was that it would be a perfect solution. So you've actually got to position what you've got, and I think that's where AgResearch have been very good with the endorsements, and this is what we've done, and this is what how it's been tested, and all the rest of it.
Excellent. More questions, please? This one here.
So, given that AR1's now, what, 25 years sort of older, that endophyte strain, and AR37's 18, are we expecting to start seeing pushback from the grubs and everything else, from the insects? Are we starting to see resistance to the toxins that are starting to come through? And are we starting to look at actually different endophyte strains now to come through on those?
Well, we've always been looking for replacements actually for AR37 and AR1, but even though we only talk about some of those key compounds, if you look at the genome of an endophyte, we know that they make at least 20 different compounds, and we are on the journey right now of discovering what some of those new compounds are. So I think one advantage of endophytes is they don't just provide the insect resistance using one mechanism, and even within like one compound. So you could imagine that you could get resistance building up quite quickly. And even within like the epoxyxanthotrim pathway, you know, they do produce intermediate compounds as well that also provide resistance. So it's not just the end products. So they're very, very complex chemistry endophytes. And I guess the other thing to remember is farmers make choices over what endophytes they want. So we still have some standard endophyte. We have, you know, a whole variety of other seed companies with their endophytes too that have different chemistry profiles. So there is some biodiversity out there, and there are some new endophytes that will be replacing AR37 and AR1 in the very near future. In fact, I think AR128 has just been softly launched, so that will be the new replacement for AR37.
Given this has been a 40-year program, what chance are there today of programs starting up and with chance finding and begun to go this long to actually return benefits?
You know, sometimes it's a lottery, right? And some people are just very lucky and they'll just find something very interesting with a very quick look. But I think we're trying to, in this case, replace AR37, which you can see the only thing it doesn't really do is it doesn't have grass grub resistance. And so that's also been a big focus for us over the years. And it's got some negative things with sometimes seeing ryegrass staggers. So, you know, it's science, good quality science does take a long time. And the reason that we've been successful is we've had concerted funding, and it's been funding that's allowed us to do that fundamental to applied. Big team of researchers, and you know, most of us, I mean, I did my PhD on endophytes in 2000, so I've been working on endophytes for 25 years. And, you know, I'm one of the younger ones here, so, you know, we have a huge amount of knowledge, which helps. Did you have something you wanted to say, David?
I think this award's been quite useful for us, but also for science, that highlighting how it can take a long time to reach the benefits of science. And yeah, look, we've been lucky. You know, Linda and I are lucky. We've been a great winning being ready because of the people that came before us. We could easily have researched all sorts of other possibilities and not being on that winning being vegan. And I can see, Trevor, you're going to say something, or Dick. This is the problem for research. Dick, did you?
Thanks, Dick.
Your first slide had the Prime Minister in it. Did you use the opportunity to lobby him about genetic engineering and encourage him to be brave?
Absolutely. So, and we've even had Honourable Dr. Simon Retty come and visit AgResearch, and we've shown him some of our gene editing. And he said to me, the one thing I can do to help you is to work on the gene tech bill. And I said, yes, that would be great, thanks. So I am quite hopeful. We have had a lot of industry lobbying, and they've really been afraid about the impact on their markets, and that the gene tech bill should really consider the market evaluation in that. However, if we look across at Australia and what they've done, we've actually had industry leading frameworks where we can still have a choice on the market. So you can have products that are being sold that are GM, that are non-GM, or an organic, because everyone wants to have a choice of technology, and it's their right to do so. So I think we can have optionality, but the gene tech bill just allows us to have the genetic technology in the toolbox.
There's a question on Chris, we come back to you, Trevor. Yeah.
Yeah, a bit related to that, I was about to ask about CRISPR and how feasible will be to apply these technologies into the field. Like for fundamental science helps a lot to understand the systems, but applying this technology into the field, how feasible will that be?
Well, at least for the endophyte, I mean, we have already used CRISPR and made a number of edits, and they are in the field in Australia. So with New Zealand's gene tech bill, we won't really know until the secondary legislation comes out as to the detail, but it's supposed to be like Australia, which means we will need to have stewardship plans in place. We'll need to be able to detect and monitor. So there will still be regulation and control, but we'll be able to sow them outside anywhere we want, essentially, but with a stewardship plan in place of how we monitor and track.
Thank you.
Trevor.
Okay. Yeah, congratulations on the presentation. I think it's a fantastic example of how young scientists need to look outside the current paradigm. I remember when I started research, people were on about plant breeding. It was all about plant breeding to get better plants for insect resistance. And it took the observation, the thinking about it, and the observation that there could be a microbe involved, there could be microbial association. And we've seen from that spark how far it went and how it's changed. And what you've achieved is a magnificent example of the plant microbiome, you know, which has suddenly become, in the last 5 years or so, has become the current paradigm, really of the importance of all our microbiomes, plant microbiome and microbiome of carpet. I think, you know, it's so that much, but everything has to be a microbiome these days. A lot of it, I think, takes the position too far and is speculative. And everything can't be controlled if you've just got a better microbiome. But this is a fantastic example of what an effective microbiome can produce for us. So, congratulations.
Thank you.
Stephen.
I just like to say how thrilled I am to hear stem wiggle mentioned so often. What I'm interested in also is some varieties of ryegrass multiflorum that are less resistant, and wondering how you're getting on with that. I will add that I see what we've got in New Zealand, quite good IPM. We've got a biocontrol that's doing a pretty good job taking pressure off those plants where endophyte isn't working as well as it could.
Yes, I should have mentioned that. I meant to have mentioned that in my reply. Yes, 100%. So it's not just endophyte in that case for Argentine stem weevil. There are other.
So you get integrated pest management.
Yes, you do.
Yeah.
You do indeed. Yeah.
And just double emphasize that we should not focus on one solution.
That's right.
And I do emphasize, as I said in my talk, the fantastic entomology work we've had in New Zealand over decades, and to understand these relationships and biocontrols as well. So definitely integrated pest management.
Sid Easton referred to the parasite as a biological curiosity.
Yeah. I suppose an illustration of that, Steve, is talking about the Italian way, annual ryegrasses. So when we put the endophytes from perennial ryegrass, when you put them into the Italian or annual ryegrasses, the alkaloid levels, so these endophyte toxins, become less.
Really?
So your control from endophyte becomes less. So it's more important that you actually control the insect in the first place. So
Pressure off.
Yeah.
Perhaps the final words.
David, in your field work, did you take any care about standardizing the nitrogen supply in your comparisons of new creations? Because it really does affect the animal behavior. If you have high nitrogen, it doesn't matter damn how many fancy endophytes you've got, they'll eat it.
Yeah. That's a good point, and the area for more research, I think. So what we did study was when you put different rates of nitrogen on, how it affects the alkaloids, because that affects insect protection and animal grazing behaviour. And there's actually quite mixed results. Some studies would say go down, some would go up. But I think in practice and in commerce, it has not been an issue in terms of animal production and persistence of pastures. But from a research point of view, it still needs some work being done there. That last word, Carol? Or yours last word?
No, it might be mine, the last word. David and Linda, thank you for an absolutely fantastic presentation. Also, huge congratulations on your the award that you won. It's absolutely great recognition of a remarkable journey, one of dedication, innovation, and when there has been real world impact in our farms here in New Zealand, but across the globe in different places as well. And many congratulations to you and the team on the impact that the work that you have done over many decades now has had on the agricultural sector. Thank you very much.
Thank you.